loud. All right. So, yesterday we talked about like an overview of cell respiration. If you've not watched the video from class yesterday, please take the time to do so within the next day or two. I promise it will really, really, really help you out. Hide that really quick. So, today we are kind of gonna get into the nitty gritty like process of cellular respiration. So it's going to be kind of doodle heavy just because it's easier to visualize when we got pictures and we can count carbons. But like I mentioned yesterday, cellular respiration happens in 3 steps. It starts with glycolysis. Then we have the Krebs cycle. And then we have an electron transport chain. Those are our 3 big steps. So we're going to go through step by step. I'll draw it out, give you a little bit more detail, and it'll be a fun time. So we're going to start with glycolysis. Glycolysis is one of those words where if you know the roots and the prefixes and the suffixes, it will tell you exactly what it means. Glycolysis, or glyco and lysis. Glyco means sugar, lysis means to break. So this is just breaking down sugars. So, during glycolysis, 1 molecule of glucose remember glucose is C6H12O6, or a 6-carbon sugar. I'm going to point out the number of carbons a lot just so we can kind of keep track of where our sugars are going, where our carbons are going, to make sure all of our math works out and we're not losing anything. So during glycolysis, 1 molecule of glucose, which is 6-carbon, is transformed into 2 molecules of pyruvic acid. Pyruvic acid is 3 carbons. So, sugar, 1 molecule of glucose is going to go into glycolysis. Glycolysis is just going to snap it in half. Yes. I'm just going to extend this really quickly so I don't have to mess with it. Okay. So, in order to get this process started, we need to use 2 ATP. Glucose is stable, that's why we like to store energy in glucose. So it takes energy to break it. But during this process, whenever we are transforming glucose, we also get 4 ATP. Which means we have a net positive of 2 ATP. And make that stand out. So 1 molecule of glucose is going in. We're going to start by spending 2 ATP to break it up. We're going to get 4 ATP out, and we're going to get pyruvic acid. Takes money to make money, essentially. Got to do the investment in order to get the reward. During this process, we remove electrons from glucose. And give them to an electron carrier. Now, our electron carrier in photosynthesis was NADP plus NADPH, right? In cellular respiration, our electron carrier is NAD plus, and it will when it gets that electron from glucose, will become NADH. This is where it gets a little alphabet soupy, because NAD plus and NADH are for cellular respiration. NADP plus and NADPH are for photosynthesis. The way I remember it is the one with the P is for photosynthesis. Photosynthesis starts with P. So if it's got a P in our electron carrier, it's for photosynthesis. If it does not, it is for cellular respiration. So we're going to draw it out. We're going to start with glucose. Remember, this happens in the cytoplasm. Our glucose, we're going to take 2 ATP, we're going to put it into the cycle, and we're going to get out 2 ADP, so we're going to spend that energy. We're going to break those phosphate bonds. We're going to get 2 3 carbons. Put a little arrow here so we know which way it's going. Then we're going to take some of those electrons. We're going to have 2 NAD pluses, and we're going to get out 2 NADHs. NADHs have energy because they have those electrons. So if you can put the little spiky cloud around it. When we get those electrons taken off of there, we're also going to get some energy. We're going to take 4 ADP, and we're going to get 4 ATP. So we've got 2 in, we're getting 4 out, so that's a net positive of 2. I give you $2, you give me $4, I have made $2. And we are left with 2 molecules of pyruvic acid. And that will go to the Krebs cycle. So our entire process of glycolysis, the first step of cell respiration, we put in 1 molecule of glucose, and we're getting out 2 ATP, 2 NADH, and 2 pyruvic acid. Two pyruvic acid, 2 ATP, and 2 NADH. There are some advantages to this process. Glycolysis is great. Like, glycolysis is one of the most common chemical pathways in life. It is super speedy. It does not take a lot of time to get this process done. It keeps up with high demand. And it does not need oxygen. But we're only getting 2 ATP out of it. We need to get as much energy as possible out of our glucose molecules to make it worth it. That is glycolysis. Does anybody have any questions? Okay. Do I want to change color? Nah. All right. So we are going to move on to the Krebs cycle. Remember, the Calvin cycle is for photosynthesis. The Krebs cycle is for cellular respiration. Step 1, we've got that pyruvic acid coming in, right? We start with pyruvic acid. We're going to break it down into carbon dioxide through a series of energy extracting reactions. Actually, hold up. Take that one off of there. The overarching idea, is that pyruvic acid is broken down into carbon dioxide in a series of energy extracting reactions. That's the gist of the Calvin cycle, or the Krebs cycle, Krebs cycle. So, if you remember our mitochondria, right? We have our outer membrane, and we have our inner membrane. This space in the inner mitochondrial membrane is called the mitochondrial matrix. So we've got our outer membrane and our inner membrane. I just want to orient you here. So it's like a compartment within a compartment. So this begins when pyruvic acid enters the mitochondria. So step 1, pyruvic acid enters the mitochondria. Give me 1 second. I want to double check something before I tell you. I don't want to tell you wrong. Okay. Want to make sure I got it right. That pyruvic acid enters the mitochondria and passes through both membranes. We are in the matrix. That's step 1. Had to double check. Step 2, our pyruvic acid is 3 carbons, right? 3. We're going to lose one of them. So we lose a CO2, so pyruvic acid loses CO2 and gains something called CoA, which is like a subgroup. So we've got pyruvic acid, we've lost a CO2, we've gained a CoA group, and this gives us something called acetyl-CoA. Now that we have this acetyl-CoA molecule, acetyl-CoA is a 2-carbon molecule. So acetyl-CoA, 2-carbon, is added to another 4-carbon. I won't make you memorize the name. I believe it's oxaloacetate, but we're gonna not make you memorize all these steps. So it's added to a 4-carbon sugar. This gives us citric acid, which is a 6-carbon. Sometimes the Krebs cycle is actually called the citric acid cycle, because citric acid is our first big production in this process. So we had our pyruvic acid, we lost the carbon dioxide, we gained a CoA molecule. That CoA helps bind to this 4C intermediate, and we get citric acid. Citric acid is a 6-carbon molecule. Now our carbon compounds are going to get broken down. Carbon compounds break down in a series of steps. CO2 is going to be released. That's where our carbon goes when we break down a carbon compound. Our energy is going to be stored temporarily. So as we're breaking down these carbons, we are releasing CO2 and capturing the energy that was in those carbon bonds in ATP, an electron carrier, and NADH, and another electron carrier called FADH2. All of these have all of our energy. So if we're tracking carbons now, all of the carbon that was in glucose is now released as carbon dioxide. And all of the energy is stored in ATP and these two electron carriers. But it's a cycle. It's a cycle because the final 4-carbon compound, after all of our carbons from glucose have been released, the final 4-carbon compound is the one that started it all. So the 4-carbon compound that acetyl-CoA joined to at the very beginning is recreated at the end of this cycle. Am I doing okay? All right, we're going to draw this out. Remember, we are in the mitochondria. In fact, we are in the inner membrane of a mitochondria. We're going to start with our pyruvic acid. Pyruvic acid is going to lose CO2, and we'll have 2 carbons. We're going to add that CoA molecule. And this is our acetyl-CoA. Remember, our acetyl-CoA is going to get added to a 4-carbon molecule. We are going to get a 6-carbon molecule. Okay. And this is citric acid. That's a little wonky, but I promise it'll be. It's going to be a cycle. I know it's a wonky circle, but what happens is we're going to lose the CO2. So we're going to get down to 5 carbons. We're going to take some NAD plus and get some NADH. Because when we're sucking off those carbon dioxides, we're also going to release some electrons. We're going to lose another CO2. We're going to take more NAD plus and get more NADH. And now we have a 4-carbon. But that 4-carbon is not the one that we need, so we're going to break it down a little bit more, get some ADP, make some ATP. And there's one more step in here where we take some FADH and get some FADH2. FADH2 is the energy one. So I know that's a little wonky. But that's the citric acid cycle. Now there's a bunch of steps in between here where we keep breaking down carbons and we keep moving stuff around. But the main gist here is that we are going to take our pyruvic acid, change it into acetyl-CoA, feed it through this cycle. We're going to release the carbon dioxides. We're going to get NADH, ATP, and FADH2. Doing okay? Okay. We have one more step. We're just going to start a new page. That step is the electron transport chain. So, this is probably my favorite step in this process. It's like the idea that life figured out a way to make this happen blows my mind. So, in the electron transport chain, we use high energy electrons from glycolysis and the Krebs cycle These are in the forms of NADH and FADH2. And our goal is to convert ADP into ATP. We've gotten some ATP from the Krebs cycle. We've gotten some ATP from glycolysis, but not nearly enough to carry out our everyday processes. Our whole goal is to break sugar down and get the energy out of it and convert that energy into a form we can use. And that form we can use is ATP. So, no, go back. Okay. NADH and FADH2 are going to pass high energy electrons to carriers embedded in the inner mitochondrial membrane. So in the inner mitochondrial membrane, if I could get this marker open, we've got proteins in here, embedded in the membrane, just like those proteins were embedded in the membrane on the thylakoids. So NADH and FADH2 pass electrons to carriers embedded in the inner mitochondrial membrane. Oh, inner only has two ends. So NADH and FADH2 hold those high energy electrons from breaking down sugars. And we're going to pass them to other carriers that are embedded in the mitochondrial membrane, the inner mitochondrial membrane. As they pass through these carriers, the carriers are going to use the energy from those electrons, the little bit that they have from each pass, to pump hydrogen ions out of the matrix. Yes. As electrons are passed through carriers, this is against the gradient or against the concentration. So when we talked about diffusion, usually things want to go from an area of high concentration to an area of low concentration. But as we are using this energy in the electron transport chain, we are going to pass things from low concentration to high concentration. We want to get all of those hydrogen ions out of the matrix. At the end of the electron transport chain, talked about this a little bit yesterday. At the end of the electron transport chain, oxygen acts as our final acceptor. It'll gain electrons and hydrogen ions, and we will get water. This is how water is a byproduct of cellular respiration. Because when oxygen gets those final electrons, we're going to reform water. And we're almost there. But now we have this gradient built up with a lot of hydrogen ions outside of the matrix and not a lot of hydrogen ions inside the matrix. H plus concentration differences creates a gradient that has potential energy. Do you remember when we talked about photosynthesis, how during the light reactions we had electron transport chains pumping hydrogen ions against their concentration gradient, and the only way they could get back to where they wanted to go was through a molecule called ATP synthase? Exact same thing happens here. Just like in photosynthesis, hydrogen ions move from high concentration to low concentration through ATP synthase. Which will then catalyze or make the reaction happen that turns ADP into ATP. This process of using the gradient through ATP synthase is called chemiosmosis. Just as a little vocab word for you. So as our electrons move through an electron transport chain, we are pumping hydrogen ions out of the matrix. But because physics, those hydrogen ions want to move into the matrix because they're really jam-packed together and they want to get the heck away from everybody else. The only door they can go through is ATP synthase. When they go through ATP synthase, it's like a water wheel. As the hydrogen ions go through, the top of that will spin and we will get ADP turned into ATP. So I'm going to draw it out. Drawing always helps. Cool. Okay. So we've got our mitochondria. We've got our inner membrane. And in here is the matrix. We have our electron transport chain. ETC. Electron transport chain. Our NADH and FADH2 from glycolysis and the Krebs cycle. It's kind of messy, but they will pass electrons to the electron transport chain. As those electrons move through, at the very end, we will get those electrons plus H plus water, or plus oxygen, and that will give us H2O. That's a little bit messy. I can redraw this if you'd like. So we've got our electrons from NADH and FADH2. They're going to be passed to the electron transport chain. At the very end of the electron transport chain, that oxygen is going to meet up with those electrons, meet up with that water, or those hydrogens, and give us water. But while this is happening, we are going to pump. We're going to say that these little circles are going to be H plus. We're going to pump H pluses out of the matrix against their concentration gradient. And somewhere else, we have our ATP synthase. Looks kind of like a T. And those hydrogen ions are going to flow through ATP synthase And we're going to get ADP into ATP. Hold on. All right. I will cover a quick synopsis again tomorrow. If you have any questions whatsoever, please let me know. There's no exit ticket to worry about. I know this is complicated stuff. Thank you for your attention. Love it.